We're going to talk about HIV and AIDS, human immunodeficiency virus. So you get an immunodeficiency. This is a virus that attacks the immune system. Acquired immune deficiency syndrome. So once you've progressed from HIV infection to having an acquired immune deficiency. So this here is once your immune system is not functioning anymore, and it's not functioning because it's been attacked by the by HIV. So in order to understand how this works, I want to kind of review how the immune system works, because you can't really understand the attack without uh, getting it fresh in your head exactly how the immune system works. Um, so I've drawn here, I've decided to go with the mucous membrane rather than the skin. Remember, on the skin, you would have to have a break in the skin, and the same thing can happen through the skin, the surface of your skin. But here I drew a mucous membrane because it's a little bit easier, and because it's a little bit easier for the HIV to get into the interstitial space here, um, I thought I'd go with that. That's where most of our HIV infections are acquired, is through the mucous membrane. Um, so I drew these orange here, the cells of the mucous membrane. That green line across the top represents the mucus lining. So underneath here, these odd-shaped guys here are dendritic cells. They are a type of white blood cell. I've also drawn a mast cell in there. Um, and so the dendritic cells, they lie right under the surface of the mucous membranes. And they also lie under the surface of our skin. So if I was talking about a cut in the skin and an infection, I would be talking about the dendritic cells, because they're just lying in wait there for any kind of pathogen to go in there. Got to remember what pathogen means, right? Patho is disease, generating disease. So it's something that generates a disease. It's a bad guy. It's a germ, right? So you've got these dendritic cells lying right under the mucous membrane. Now you get a pathogen on here, could be AIDS, whatever, you know. And also you got to remember, it could also be an allergen. Sometimes it's not necessarily a bad guy. It's just something that your system doesn't seem to like, so it will do the same thing, unfortunately. So these guys here, the thing about the dendritic cells is they will actually reach up through the surface of the mucous membrane and grab a pathogen when it's out there and pull it under, right? Doesn't do that on our skin, right? If I get a pathogen on my skin, that just serves as a barrier. It doesn't pull it in, right? But inside in your mucous membranes, it does. So it pulls it in here, and then it engulfs it, right? It phagocytizes it, chops it up, and takes a part of it and one of its marker parts and presents it on his back. And then we'll call them presenter cells a lot of times when they talk about them going down to the lymph node. So this guy here is going to come, and it's going to come in here. Right now it's got that pathogen on its back, and it's going to float through this lymphatic. This is supposed to be a lymphatic vessel, and this down here is a blood vessel. It's going to float through the lymphatics to the nearest lymph node. And I've got a lymph node drawn right here. And another thing is going to happen too, when it comes through here and it's activated now because it found a bad guy, it releases a chemical, and it will cause this mast cell to release histamine. Um, and what histamine is going to do is it's going to cause vasodilation of your vessel here, right? So your blood vessel now is going to be dilated, and it's going to make that blood vessel leaky, right? Because in between all of the cells of the blood vessel, when it gets dilated out like that, the gaps between them get larger. And so now the plasma here, more plasma is going to be able to leak out of the blood vessel into the area. There's a good reason for that I'll get back to. So now this dendritic cell gets down here into the lymph node, and it presents itself. It's got this guy, right? It's got this bad guy, chopped him all up, put a chunk of him on his back, and went down to the nearest lymph node. The lymph node down here, you've got a lot of different white blood cells in here, but you've got your T cells and B cells. There's T4s and T8s. A lot of times they'll call these T cells CD4 cells. And I'm just bringing that up because that's what we'll call it when we're talking about HIV infections. So anyway, These guys come down here, and we have a lot of different T and B cells that have come across uh, pathogens, right? And a lot of these are memory cells, and they will remember if this pathogen has ever been there. So if this pathogen happens to have a T and B cell down here that remember it, that will line up with it, and they remember it right away, there's going to be a lot of them in the system. Boom, it's going to activate, it'll kill that sucker, and it will release all of these other ones, right? It will start making more of them, and all of the B cells that match that pathogen are going to come up here, and now you've got vasodilation that happened from the histamine. So you got this vasodilation, and they're all going to collect in here, right? There's going to be an increased circulation to the area because of that vasodilation. And because of the increased leakiness, these white blood cells, T and B cells, are gonna leak out into the interstitial space and be all over here to wherever that these bad guys are. Right, they're right there waiting. If I cut my finger, it's going to be all swollen there right now, and it's going to have a lot of inflammation in the area. That's what I'm actually describing as inflammation. Um, and so now you got increased fluid in here, increased fluid comes out through here, and you get a increase in your mucus, and it's more watery because you've got all this fluid coming from the plasma. So you get it right. If you can get a runny nose, you're going to flush this out. So whatever, if there's more of these guys stuck in that mucous membrane, boom, you're flushing them out, right? If it's in your eye, you're going to get a really itchy, tearing eye. If it's in your upper airway, right, you're going to get a runny nose and stuff. So that's what happens, and this is how this whole thing takes place. You've got your B cells and T cells here. You got your dendritic cell that carries this down here. So when I get talking about this HIV stuff, you'll know what I'm talking about, right? When I say a presenter cell, I just mean one of these guys here that's presenting down here. Right? When I say a dendritic cell, I'm talking about one of these guys grabbing a hold of it, right? The same cell, actually. Just that down here, they call it a presenter cell because it's taken it, put it on its surface, and went down there and presented it. I like to think of the lymph nodes as like the police station. There's a whole bunch of cops there just waiting, and you come and say, hey, here's the bad guy. It's a wanted poster. Boom. They're all out there, and they're all going to go get him. So if they recognize him, they're going to get him just like that. Let's start out by just generally talking about what happens in an HIV infection. HIV likes to infect the T4 white blood cell. The T4 white blood cell is also called a CD4 because it has on it a CD4 receptor site, and so they call it that. Now there's also a T8, right, which is the CD8 on it. So this here, that the CD4 receptor site on it, these guys are also called T helper cells, right? They call them T helper cells. They call them the 8 T killer cells. And there's good reason for both of those nicknames, but the T helper cells is a super important cell in our immune system because that's why they call them helper cells, because those suckers help everybody else, right? A B lymphocyte cannot kill anything without a helper cell coming in and ensuring that, yes, this guy is not self, this guy is somebody that we can attack. And that's, there's another whole video on how that all happens. But right now, just it's important to remember that the CD4 cells do more than just attack lymphocytes on their own. They also help the rest of the immune system function. Basically, what happens is we have our HIV virus picked up by the dendritic cell. The dendritic cell takes it to the lymph node. In the lymph node, we end up with one of these HIVs getting into a CD4 cell, and I will talk about how all that happens. So it infects the CD4 cell and ends up killing off CD4 cells. So once it gets into the CD4 cell, though, it will use that cell to reproduce, and it will just start reproducing like crazy. And we'll talk about that process too here in a few minutes. But once it gets going on the okay, so when we have an acute HIV attack, so the acute stage would last about a month. And that is where the CD T helper cells get infected. And they get infected because they are brought in by the dendritic cells, right? The presenter cells. Some intact HIVs can hitch a ride on that dendritic cell. It's going to take them right into the lymph node, and the lymph node is where there's lots of these T cells. So once it gets in the lymph node, it will infect one of the T lymphocytes. A CD4 cell, as a matter of fact, that's what it prefers to attack or to get into. So with the T4 cell, it's also called a T helper cell, and the reason they call it that is because it helps everybody else in the immune system. A B cell can't even kill anything without the T helper cell approving it, right? So, and there's kind of a whole video on that too. So, what we end up with is high viral loads. And a viral load is how much of the virus is in your system right now. And that's your viral load. So here in the acute phase, you get a really high viral load because they start reproducing like crazy in all of these T cells. And then these, and also they also call these T cells CD4 cells. And the reason they call them that is because they have a receptor on it that's a CD4 receptor. Right, so you can hear this same cell referred to as a T helper cell, a T4 cell, or a CD4 cell. They're all the same thing. Let's see. And so what happens is these CD4 cells or T cells, and you just got to get used to the fact because I'll be using all three of those terms interchangeably, and I'm talking about the same thing though. You need to know that. So what happens is these CD4 cells get the viruses inside them. They detect that they have the virus inside of them. They end up killing themselves, right? Suicide, boom, right? Get rid of myself because I'm infected with something that I can't get rid of, and I don't want it to go any further than this, so I'm killing myself. That's called apoptosis. All cells have the ability of apoptosis, and they will carry it out when the time is right, when the cell believes the time is right. Another one is pyroptosis, and that is a result of inflammation. If you get intercellular inflammation, which you will get, right? You can get with this, and it will kill itself. So anyway, the T cells, the CD4 cell count is getting lower, and that's why I've kind of switched from calling them T4 cells or T helper cells, and I'm kind of going to start calling them CD4 cells because that is what they're usually referred to when we're talking about HIV. And the reason is because that receptor is, the CD4 receptor is super important for the HIV cell procedure of getting into the cell. It actually attaches to that CD4 cell. So what you end up with in the acute phase is a real high viral load and a dropping CD4 count, right? Until seroconversion takes place. And seroconversion, that means that you have developed the antibody, and it's now at a level that you can detect the antibody for that specific virus. So once you can test the blood and you can find that, yes, they have the antibody, the HIV antibody, then that means seroconversion has taken place. Usually takes, I don't know, starts in a couple of weeks, but usually you can detect it in about a month. Now, when this has taken place and you have this viral infection, sometimes the symptoms are really mild. They just feel like they have a cold, but they can also feel pretty sick because this is the virus that they haven't had before. And usually when you are going through seroconversion and you are building up that immunity, you feel pretty bad, and it can last a while. Like I said, it takes 2 weeks to a month before you're going to be seroconverted. So you'll feel pretty sick during that time. Usually you'll have fever, you have fatigue, body aches, rash, swollen lymph nodes, because this is all going on in the lymph nodes, and just flu-like symptoms. So now after that acute phase, you go into a latency period, and this can last 2 to 10 years. And this time the CD4 cells are recovering a little bit, they're reproducing, the immune system has come up with a response, and they are out there trying to kill them, and I'll talk about that a little bit later. But so now you've got a fight of, is the HIV reproducing faster, or is the immune system able to kill HIV faster? Well, unfortunately, what's usually happening here is HIV is reproducing a little bit faster than what the immune system can fight it. So now think about it. If it's winning just a little bit, it's fighting the immune system, right? It's attacking T4 cells. So the fact that it's attacking those important immune cells means that as it wins more and more, the immune system gets weaker and weaker, right? So it will gradually take over. And that's what happens during the latency period. And like I said, it can last 2 to 10 years, depending on a lot of different variables. You'll get that high viral load, and then that viral load will start dropping as the immune system starts killing them off, and it kind of hits this spot here, and it goes kind of even for a little while, but slowly that viral load will increase as it slowly starts to win over the immune system. So in the acute phase, they had these symptoms of fever and body aches, and rash, all these flu-like symptoms. Usually during the latency period, they don't really feel too bad. It doesn't usually take much for them to feel bad, though. They might get some sore throats, things like that. But as the viral load rises and the CD4 count drops, they will develop more and more symptoms. And they can get to the point where they have acquired the AIDS syndrome, so the acute immunodeficiency syndrome. So once that has taken place, that means that they don't have an immune system left anymore, right? They've gotten to the point where their immune system is just not functioning. And we'll talk about exactly what it takes to be diagnosed with AIDS. But definitely know this: it is the infections, secondary infections, that usually kill the person with AIDS. And then you might say, well, how do you pass AIDS on? Well, it's passed from one person to another. And how contagious a person is directly related to how high is their viral load. Their viral load is low, they're not that contagious. Their viral load is very high, they are very contagious. Those are the people that you don't want to come in contact with their body fluids. Okay, transmission of body fluids is how this is transmitted. It can go across broken skin, it can go directly into the blood, or across mucous membranes, all the way from the mouth to the anus, right? It can enter through there in those mucous membranes. The respiratory tract, under the eyelids, right? All the same places that you can get any pathogen can enter. Often it is transmitted in semen or pre-seminal fluid, vaginal fluids that can be transmitted in, and fluids in the rectum. And down in the rectum, right, you actually have a lot of fluid there that has been going into your colon, and by the time you get down into the rectum, your system is supposed to try to reabsorb most of that out so that you don't lose all your fluid out through the anus. But sometimes it doesn't. But anyway, you have fluid there, and that fluid can carry HIV in an infected person. Anal sex is the highest risk of transmission. Can be transmitted to the recipient, whoever gets the fluid put in them. It can also cross the membranes at the tip of the penis. So it can go from the rectal fluids into the person's penis, and it can go from, of course, from the semen and be absorbed through the mucous membrane in the rectum. Now, I say that it's the highest risk, and you might start looking in some literature and you'll see that there have been more people in the United States that have gotten it through vaginal sex. Well, they collect HIV through vaginal sex, but there's a lot more people engaging in vaginal sex. But the highest risk, percentage-wise, by far, is anal sex. Oral sex of any kind, right? Oral sex of any kind, you can transmit HIV. Um, because you are mucous membrane to mucous membrane. You've got to remember too that the tip of the penis is the beginning of a mucous membrane, although that's not very much that's being exposed, so the risk is a lot lower to the male in something like that than it is to the female, well, to the person giving oral sex has got a lot more risk than the person receiving oral sex. I guess I'm trying to say this as tactful as possible. Okay, so infected blood. You can get it in infected blood. IV drug uses, sharing needles. They pass it from one person to another. Healthcare worker accidental needle sticks. It's another way that you can end up getting it. Blood transfusions, although that I mention it, but it's something that doesn't really happen much. They have been really strict about checking it for HIV virus since, God, I don't know, like the late 80s, early 90s, and that time period. And an infected mother can give it to her child in childbirth. You got to remember, children, they haven't really built up the horny layer of keratinized cells in the surface of their skin yet. So it's almost like they have mucous membranes everywhere. Just things get absorbed so easily that they're primed to pick that up during childbirth. So a lot of an infected mother, you got to be very careful to try to avoid trauma during childbirth. And HIV can be transmitted through breast milk. Of course, a break in the skin, HIV can enter through the break in the skin. It can get into the interstitial spaces, get picked up by dendritic cells, carried into the lymph nodes, or it can get into the blood and go directly into the lymph nodes or direct contact with CD4 cells that way. The pathogen in the mucous membranes can be grabbed and pulled right in through by dendritic cells, right? Chops it up, presents it on its back, goes to the nearest lymph node. T and B cells will look at it, right? And some of these T4 cells, they have this CD4 co-receptor, which will interact with the antigen-presenting dendritic cells, right? That's what CD4s do. They're T helper cells. Anyway, the dendritic cell presenting this way is often called an antigen-presenting cell. This is all normal. This is the way it's supposed to happen, right? Because I was talking about when a pathogen comes in, so everything sounds like normal. But in this case of HIV, a virus can piggyback on it. Now I'm going to go here to the board and kind of show you how that happens. Okay, so here I've drawn a lymph node, right? And of course, I've only got one cell here, but there would be thousands. So we've got this T cell here, this T4 cell, and it has a CD4 receptor site on it. It would have hundreds on this cell, right? Not just one. Now this here is the dendritic cell coming in, and it has the piece that it's going to present, but it also has been hitchhiked. Somebody hitchhiked here, so he hitched a ride on this T4 cell and came in. So we've got an intact HIV coming in. Okay, so we've got this HIV that is piggybacked a ride in on this dendritic cell, and so now it gets released off of here and it comes on over here and attaches to the CD4 because this here has a receptor on it. It's called a GP120 receptor, and it adheres to the CD4 receptor. So then you get this over here, right? It's coming right here. You've got your cell here now because it is just attached to it. And so now it's attached to this, but it still can't get inside, right? This T4 cell is really good at keeping things out. It doesn't want these bad guys getting in there. The HIV has another receptor, right? This is the GP120 receptor. GP120 receptor attaches here. Then it has another receptor, which is a GP41 receptor, and this guy is right here. And it attaches, it adheres real well to the co-receptor of the CD4, which is a CCR5, right? So it attaches to the CCR5 receptor there. So now it's got a double attachment, right? When it was attached just to the CD4, it was not stable enough. But now that it's attached in two places, it's stable enough to bore a hole right through here. And it bores that hole, and now it can release its genetic materials down into the T cell. And that's where the infection starts. Now, the interesting fact is there are people who have a mutation that causes a mutation of this CCR5 receptor, and so then the co-receptor up here in the HIV cannot attach to it. So the fact that it can't attach to it means that it can't get its genetic material into the T cell, which means it cannot infect the T cell. Those people don't get AIDS. They found this way back in the late 80s when they were doing a study, and they're trying, AIDS was pretty new, and it was very devastating, and it was killing everybody, and they found that in this brothel down in the middle of AIDS territory in Africa, that there were a couple of prostitutes there that weren't getting AIDS. All their friends and stuff kept dying around them, and they just did this for years and were not getting AIDS. So they did a big study on them, and they looked at everything in their blood, and what they found was that in their T cells they had this mutated CCR co-receptor. The CCR5 is a co-receptor of the CD4. So with that mutation, the AIDS wasn't able to get in there. We also have medicines that do a good job on this. One of them, they're called infusion inhibitors, and what they do is they attach to this receptor on the HIV, the GP120 receptor, and by attaching to it, it is free to attach to the CD4 receptor on the T cell. So that will stop it from attaching to it. Now, there's a whole lot of these receptors on each one of these cells. I only drew one, but there's hundreds and hundreds of them. So they don't necessarily block them all, but they make it a lot less likely that it's going to end up attaching to that. And because they don't block them all, we can give them another kind of medicine too. And what this medicine does is it attaches to the CCR5 receptor on the T cell. So when this guy comes around, the HIV, if it gets attached to this, well, it can't get that second hold on it, so it can't drill the hole in there, and it can't inject its genetic material in there. Now, like I was just saying, there's lots of receptors on there, so even with these medicines, they can end up attaching and getting into the cell. Doesn't block 100% of them, but it dramatically decreases the amount of HIV that is able to get into the T cells. And since it doesn't block them all, we'll put them on more than one medicine, right? We'll stop them later on through the process of infection. So we'll keep talking about what happens here. So once inside the cell, all viruses will use the organelles of the cell to survive, and they'll make copies of itself, and that's just what viruses do. HIV gets into our DNA. And it gets into our DNA, it has some of what it sends in here, right? It sends in some RNA. It sends in an enzyme called reverse transcriptase. Reverse transcriptase. And And this is an enzyme that actually comes in here and attaches to your RNA, little divisions there, and it looks for peptides and peptides in the cell here and grabs them, pulls them in so that it can use this RNA as a template to create a strand of DNA. All right, so it creates a strand of DNA, and then that strand of DNA, it's a single strand now. Full double strand is what a DNA takes to pass on its genetic codes. So this strand here is going to automatically attach, pick up the things that it needs, right? Because just by the way DNA works, it will attach the other side of that ladder and it will pull it into it, and it creates a double strand of DNA. So now we have double strand of DNA in the cell from the virus. So that is the entire genetic code of everything that it needs in order to reproduce itself. But it doesn't really have the ability to do that yet. It has to be picked up by an enzyme called integrase. Integrase, let's write it over here, I-N-T-E-G-R-A-S-E, right? And integrase is going to pick this guy up and drag it in here. And it's going to take, you got your own DNA in here, it's going to take this little piece here and it's going to snip a chunk out of here, and it's going to insert this chunk of HIV DNA right into our own this T4 cells DNA. And something worth mentioning at this point is, as this thing is doing its transcribing with the reverse transcriptase, it doesn't do a real good job. It kind of screws up a lot. And by doing that, it creates a lot of mutations. And by doing that, this thing evolves and mutates to the point where we can't come up with a vaccine, right, an immunization that will continue to work on it, because it keeps changing its look that often. And now what happens is our body, like it does, it takes the DNA and uses that as the template to switch into RNA, and then the RNA will pull together the peptides to make the proteins that we need. Well, the problem is now in its genetic code we start making the proteins that the HIV needs, not that we need. So first thing it's going to do is it's going to put together an envelope, right? The envelope is now on a cell. We have the cell membrane. The envelope is what holds the genetic contents, what holds everything together for that virus. So by doing that, it makes this envelope. And I'm going to stick to blue, right? So it's going to have this little part of the envelope there, and then on the envelope, it has the receptors required, of course. So then they will migrate out to the edge here, right? And you're going to get a whole bunch of them here. Right, this thing is just cranking them out right and left, and these envelopes are going out there. But what else does the virus need? What else does that HIV need? Right? Well, it's the things that we just talked about, right? It's going to need reverse transcriptase. It's going to need integrase, right? It's going to need those two things in order to create a new one that can go out and create new ones. So what it does is it makes those, but it makes them together, right? You got these guys here, and they call it a polyprotein because it's got both of them and actually some other things in it too on this big, it's a long strip, right? But it's not separate, so they aren't able to do the things that they do, which is probably a good thing while it's out here in the cytoplasm. You want to get it into a new cell so it can do its thing there. Well, as these envelopes, these viral envelopes come out here, they end up budding out, and you get this guy in here. right? Right, you got the two parts to it right there that I'm going to draw anyway. The reverse transcriptase and the integrase there together in that one poly protein. And another thing that it makes in is a protease, a special protease, kind of like a scissors, and it comes in here too. And what it does is it cleaves that and it cuts that up in the pieces that it needs it to be. So instead of being big long strips of those things, it cuts them up so that now you have reverse transcriptase and integrase in there. And now this thing is ready to bud off. It'll bud off. And now you've got a new HIV cell that's got all of those things in it that it will need when it comes in. It also has the receptor on there, right? So that it can grab a hold onto another T cell, which there's plenty of them here in the lymph node. So it can get onto another T cell and infect that T cell. So now you've got this happening all around here, right? It's budding out all over this poor cell and creating all of these baby T cells, or I'm sorry, creating all of these baby HIVs that are going to go out there and do this all over again to new T cells. I want to talk about a couple of medicines. We have NRTs, nucleoside reverse transcriptase inhibitors. So the reverse transcriptase, which is what makes it so that the cell can do this kind of backwards thing here by taking the RNA and making DNA. It comes in here, and what it does is it blocks it. What it does is we in the medicine that we give, this reverse or this nucleoside reverse transcriptase inhibitor, it has like a fake nucleoside on it that will come in here and it will attach to one of these guys, and it doesn't allow anything else to be attached to it adjacently, so it stops the progress of transcribing over into the DNA from the RNA. So that's how that works. And then there's another one that's called a non-nucleoside reverse transcriptase inhibitor, and what that means is it doesn't have a nucleoside on it to come and attach here. It just has a gummy substance that likes to stick to these guys. It won't be able to do its job of creating the new DNA by attaching nucleosides to it. So I talked now, after this is made, integrase comes in here and it integrates the newly formed DNA into the our own, or the cell's DNA. So we have another medicine that does this, stopping the reverse transcriptase, and it's called Truvada. No, it's made up of two different medicines of these two different types of blocking, but Truvada is being used as a preventive medicine. And as long as the people take it every day and they keep the level of Truvada high enough that they keep enough of this in, and these are people who don't have HIV, these things are 95 to 99% effective in stopping the people from getting an HIV infection. That's pretty impressive. So by taking this every day, what it does is you've got this high level of stuff, and you keep, anytime you get in one of these RNAs, you get this infection, boom. You've already got that medicine there, and it stops it. If these people are not compliant, that 99% drops way down into the 80s. So they really need to be compliant and take these pills every day to keep that level of medication high in their system, so that any time that the infection, that one of these HIVs gets into the system and it gets in here, it will not be able to reproduce and will not be able to cause an ongoing infection in the person. But that's Truvada. It has a generic name, but I don't remember what it is. I'm sorry. Maybe you can look that up. I'll probably talk about it later when we go over slides. But they will use Truvada on high-risk people, right? People who are engaging in activities that they could very likely end up with an HIV infection. And like I said, it's 95 to 99% effective in preventing HIV infection. Now I talked about the HIV DNA, and here it is harmless, can't do anything. But once integrase gets a hold of it and integrates it into the DNA, the cell's DNA, it will start reproducing and making all this stuff. Well, there's another medicine called integrase inhibitors, right? This class of medicine, what do you think they do? Well, they block integrase's ability to break up the DNA. So that's another medicine that we work. Now, when these people are on 2 or 3 of these medicines, it really gives them a longevity of life that people with HIV infections did not have before. Now we got one more medicine to talk about, or actually I should say one more class of medicines to talk about, and that is protease inhibitors. What do you think they do? They inhibit protease. And proteases, we have this special protease that will break up this um, polyprotein down here into your reverse transcriptase and your integrase. And by stopping the protease from doing that, it will not be able to go to another cell and carry out these activities because it's going to have this useless polyprotein instead of the 2 enzymes that it needs. Now remember, our system does have a defense against this infection, right? It fights it and it does a fairly decent job for 2 to 10 years, but over time it will always lose. But with the effect, or with the assistance of these medications, all these different medication classes that I've talked about, a person can go on and live a long life. It's made a really big difference, and especially with that Truvada, if the person in a high-risk lifestyle takes Truvada every day, they have pretty much cut out the chances that they are going to get AIDS. And I said pretty much because nothing is ever 100%. But along with these infections, the way our system fights it, we have T8 cells that detect and kill the infected cells. So that's one thing that we do. We also have the cell itself, which detects the invasion and becomes inflamed. The inflammation calls for more immune cells to come to the area, and unfortunately it can lead to pyroptosis, which means it explodes. Now, I picture this as a violent explosion because the way it's described in the journal that I read, these particles fly out and they end up infecting all of the nearby, or actually not infecting, because these are particles, these are inflammatory messengers, right? Interleukins, I think they're interleukin 2B or something like that, doesn't really matter, but it's an interleukin and it goes, and interleukins are inflammatory messengers. So you are throwing inflammatory messengers right into these other T4 cells or CD4 cells, and so they get inflamed. Not because they're infected, but because they have inflammatory messengers on them, and that's what inflammatory messengers do, is they start the inflammatory process. So they get thrown into them, and they get inflamed, and then they go through pyroptosis, and then they throw their inflammatory messengers. So this chain reaction is what kills most of our T cells. Most of the T cells, when we have a low CD4 count, it's not usually caused because all of the cells that were killed were infected. They weren't, but they die from this chain reaction of inflammatory messengers getting thrown into other ones, and then they go through the process and they all end up with pyroptosis, which means that they are killed as a response to the inflammation, right? It's like, this is inflammation out of control, I'm going to blow myself up, right? And that's what they do. And also you have the apoptosis, and that isn't a response to an inflammation. That's just the cell itself saying, hey, wait a minute, I got something in me and I've been trying to control it, but it's doing some weird stuff in here. I don't want this to go any further than me, so I'm going to kill myself right here. And so that's what apoptosis is. So a person is diagnosed with HIV when they've had an antibody test that shows that yes, they have the antibody to HIV, so now we know that they have HIV in the system. Now, in order to be diagnosed with AIDS, that means that they have progressed from just having this infection to having a shutdown of their immune system. And once they have, and the way that they diagnose that, AIDS, is the person has a CD4 count less than 200.
Yeah, less than 200. So if the CD4 count is less than 200, they'll be diagnosed with AIDS. Because if they're that low, normal is 800 to 1200, right? So right around 1,000. So if they're way down less than 200, they have no immune system left, or they've acquired an opportunistic infection. That's an infection that you wouldn't get unless you had no immune system left, right? And those are usually like the fungal infections, like of the lungs, cryptococcal infection, um, a pneumo pneumocystis, pneumonia, right? If you get one of these infections, right, you don't get them unless there's something really wrong with the immune system. So if you have one of these opportunistic infections, you're diagnosed with AIDS, or if you have a CD4 count less than 200. So the things that you watch is you watch the viral load and you watch the CD4 count. If the CD4 count, usually the CD4 count is going to be dropping as the viral load goes higher, right? Because as it drops, you're not fighting as well. You're not able to fight the virus as well. So the viral load tells you how infectious the person is, right? It gives you a good idea as to how infectious they are. So those are the two labs that you're really going to be watching: is your CD4 count and your viral load. And hopefully, your viral load is actually counting up the virus. How much virus do you have in your system?